Hi, how are you? This is Ahmad Aldemiri, and I'm going to talk about the transfer of Escherichia coli to spinach by housefly. This is my second presentation for this course. The author is Justin Talley. He got his bachelor degree from West Texas in 2000, and then master degree from the West Texas again, 2001, and he also has PhD from Kansas State University 2006. He is associate professor in Department of Entomology and Plant Pathology, Oklahoma State University. For his research interests include the stable fly, biology, integrated control, and insecticide efficacy, and sampling strategies, and development of economic thresholds, and also parasite house interactions. His professional activity Entomological Society of America and Society of Southwestern Entomologists. Introduction. There are many possible routes that lead to the contamination of fresh fruit and vegetables by organisms that are associated with foodborne illnesses. Houseflies have been reported as mechanical vectors of many enterobacteria pathogens such as E. coli. Houseflies carry E. coli between animals and to the neighboring environment. Kobayashi showed that after houseflies were exposed to E. coli lawns, the bacteria could multiply within the pseudotrachea of the fly labellum. Sasaki found that when housefly fed E. coli lawns and then allowed to contact different foods were able to disseminate bacteria into food surfaces by fecal excretion for 24 hours. Objective of the study. The objective of the study included the documentation of E. coli survival and multiplication on the spinach phylloplain after fly regurgitation, and the retention of viable bacteria on fly body surfaces. Material and method. Housefly colonies. Housefly from the Oklahoma State University were isolated in a special environment, and temperature range 25 to 28 Celsius and a 12-hour high dark photo period. 2 to 5 day old adult flies were collected and transported to laboratory for all experiments. E. coli cultures and inoculating a loop of E. coli strain ATCC Shiga toxin negative with a plasmid inserts containing genes for green fluorescent protein and ampicillin resistance. The culture was harvested by centrifugation, and the pellet was suspended in sterile distilled water. The bacterial suspension was mixed 1 to 1 with manure, resulting in a final titer of approximately 1 to 2 of 1007 of manure. Growth and maintenance of plants. Spinach was grown in a greenhouse. Plants with 8 to 9 mature leaves were used to all or in all studies. E. coli acquisition sources. Housefly were exposed to 4 different um, acquisition sources. E. coli from autoclaves manure, E. coli from plate, sterile water, autoclaved manure, and algae plant. 40 to 50 housefly were placed in each acquisition source plate. After exposure, flies were um, anesthetized by pumping CO2 for 10 seconds into the exposure chamber via a port cut into the lid of the petri dish. Anesthetized flies were immediately transferred into growth chambers containing spinach plants. Relative quantification of E. coli in housefly regurgitation spots. Regurgitation spots will proceed for relative quantification of E. coli DNA using quantitative PCR. Quantification of E. coli in manually spotted droplet on spinach leaves using relative qPCR. This experiment served as a control to determine the resource of E. coli on spinach leaf surfaces without the influence of housefly or their regurgitation. Quantification of E. coli on external surfaces of houseflies by microbiological methods. The study was conducted to evaluate the persistence and colonization of E. coli on the external body surfaces of houseflies exposed to different E. coli acquisition sources. Scanning electron microscopy of regurgitation spots and fly body part. Regurgitation spots on spinach on day 0.75, 4, and 8, and spinach leaves spotted with E. coli were collected after exposure to flies on day 0, 2, 6, and 12. The labia and tertia of the flies exposed to the 4 different acquisition sources were prepared for scanning electron microscopy using the tissue fixation procedure. Result: relative quantitation of E. coli in housefly regurgitation spots. There were significant difference in the relative amount of E. coli amplicon between treatments. Regurgitation spots to deposited by flies exposed to autoclaved manure mixed with E. coli showed an 18-fold increase in the EAA amplification from days 0.75 to 4, which suggests that the number of E. coli cells increased on spinach leaves within that time period. The regurgitation spots obtained from flies exposed to E. coli lawns, the EAA amplicon levels remain fairly level and showed no significant difference in the fold change from day 0.75, 4, and 8. Quantification of E. coli in manually spotted droplet on spinach leaves using relative qPCR. This result shows that the level of the EAA amplicon and the number of bacteria did not change over time. SEM examination of regurgitation spots. The regurgitation spots on E. coli manure exposed flies contained regurgitated organic matter. It means the flies consumed from autoclaved manure. The regurgitation spots from the PP treatment also contained of potential cell division of bacteria-like organisms, but this or their size was smaller and the matrix varied in appearance from the EM treatment. Figure 1. This is Figure 1, scanning electronic microscopy of housefly. Regurgitation spots deposited on spinach leaves after feeding on Escherichia coli plates. SEM of manually spotted bacteria. On day 0, SEM of the E. coli spotted leaf surfaces showed many BLOs resembling E. coli. This is Figure 2 that shows scanning electron microscopy of control spot, manually spotted Escherichia coli or E. coli on spinach leaves. SEM of spinach leaf surfaces spotted with sterile water. Negative control showed a few slender rod-shaped BLOs that were morphologically different from E. coli. Microbiological recovery of bacteria from fly mouthpart and teres. The number of positives was high in the EM and EP exposed flies and low in the SM or LB negative control flies. When samples were retested by endpoint PCR for the presence of the EA gene, all bacteria recovered from the SM or LB exposed flies were negative for the presence of E. coli. SEM of fly teres exposed to E. coli lawns showed rod-shaped blows on the sticky hairs of the tarsal palvula. Number of BLOs were visually higher than those of flies from the EM treatment. EM exposed flies had only a few BLOs on the sticky hairs of the palvula and on the tarsus. Figure 3. This is Figure 3, scanning electron microscopic of fly body parts. Discussion. In this study, the researchers shows that flies retain live bacteria for contamination via casual contact. Their results suggest that regurgitation may deposit viable cells on plant surfaces that are capable of replication. They discovered that the plasmid carrying the GFP insert was lost fairly quickly in the absence of ampicillin selection, pressure, and plant and insect surfaces. Unexpected regurgitation spots from flies that were fed on control excretion sources showed no significant differences in food change, although there was a slightly application of EA at 4 and 8 days post deposition. The study provides the first investigation of housefly regurgitation spots on spinach leaves by SEM and documents the survival and replication of E. coli within those spots on the leaf surfaces. Mitra reported that spotting of a cocktail of 5 E. coli strain on the abaxial surface of spinach leaves resulted in bacteria survival on the phyllo plant for 14 days post inoculation. Okay, they also documented that the bacteria tighter and colonization of leaf surfaces increased over time. In contrast, the EA target of manually spotted E. coli used in the study did not go up over time. Uh, there is the EAE target DNA in the EM regurgitation spots increased dramatically in 4 days. This suggests that fly regurgitation spots serve as a nutrient source for bacteria, allowing them to survive and multiply on the spinach phyllo For SEM, the results from this research and the Sacontason study suggest that bacteria associated with fly feed could contaminate plant surfaces. In addition to regurgitation, fly body has sticky glandular hairs on the palvilla and the pseudotrachea on the labellum are additional niches where bacteria can survive. This is my reference or the article reference, and thank you so much, and please ask me if you have any question. Thanks a lot.